Thanks everyone for being here for ESPM's seminar this week. It is my pleasure to introduce Dr. Rebecca Albright, who is here with us from the California Academy of Sciences. Rebecca is the curator of invertebrate zoology and Patterson Scholar. And she grew up in a landlocked state, but fell in love with ocean ecosystems and did her undergrad at Duke University and quickly decided to do a study abroad in Australia, which further confirmed that interest and got her diving in marine systems. And then spent 2 years in the Dominican Republic working for Reef Check, which is an organization that surveys reef health in different areas. And then did her PhD in marine biology and fisheries at the University of Miami's Rosenstiel School for Marine and Atmospheric Sciences. And that actually is where I was lucky enough to get to meet her and go in the field and do some work with her. And then did a postdoc at the Carnegie Institute, again, working in Australia, and has done some really amazingly like out of the box experimental thinking in terms of trying to understand how ocean acidification has impacted reefs and with implications for the future. And today runs, does a lot of important outreach and research at California Academy, including running the coral spawning lab, which I assume we will get to hear at least a part about for today's talk. So thank you very much for being here. And in case I forget to say it later, don't forget there will also be a social hour following this in the courtyard after Dr. Albright's talk. So thank you.
Thanks so much. Thank you all for coming. Yes, it's kind of crazy that I've known Adrian for almost 20 years, it's 18 years. And we met originally paddling around in Panama in a Gulf collecting discrete water samples for carbonate chemistry. They didn't have sensors back then, so we just paddled around in literally an inflatable you would buy from Walmart and took them every 2 hours over the course of like 48 hours or something, and then shipped them back. But yeah, thank you for coming. I'm a curator at the California Academy of Sciences. I have been there about 7 years. I was previously a postdoc at Stanford at the Carnegie Institution for Science. And so I've been studying various aspects of coral reefs for the better part of 20 years. And I have the strongest backgrounds in ocean acidification, which I'm actually not going to talk about today, but given that this is like a management and policy related department, I would love to maybe come back sometime and speak to that work, because I have done a lot of work looking at the implications of alkalinity enhancement, ocean alkalinity enhancement. We're actually writing a book chapter right now for a best practices guide, and we published a paper on a policy tool for managing and mitigating OA impacts in coastal environments. And so maybe we can talk about that another time. But today I was going to focus on, I also have a background in coral reproduction. And so in a time, I've been very lucky at the Cal Academy. For the majority of my career, I kind of focused on the impacts of global climate change on coral reefs. And there was not a good story, you can imagine, kind of out of any of those experiments or studies. And since being at the Academy, I've been able to do a lot more solutions focused work, which has brought a lot of optimism and a lot of hope to my life. And that's kind of where my focus is a lot of the time right now. And so I really focus on recovery processes. We'll talk a little bit today about the plight of coral reefs. I'm sure you've seen it in the media and the news. We'll talk about why that's happening. And some of the solutions and kind of the small role that we're trying to play in that process, which has to do with spawning corals and trying to support these recovery processes, which really are reproduction and growth in the middle of Golden Park in San Francisco. So when you think about coral reefs, something like this probably comes to mind. You think about their incredibly iconic ecosystems. You probably think of things teeming with color and life in very tropical warm places. You might think of some charismatic megafauna
like maybe a sea turtle or a shark, Nemo, some brightly colored corals, things like that. Or you might just want to go on a really beautiful tropical vacation to a wonderful place and go snorkeling. And in developed countries, we primarily appreciate coral reefs for their aesthetics. But in developing nations and a lot of places around the world, they offer us very, very tangible ecosystem services that are valued at trillions of dollars a year. So they cover less than 1% of the ocean floor, but they support 25% of biodiversity, and they support the livelihoods of 600 million people throughout the entire world. They also provide us with coastal protection, so it's a human security issue at the end of the day. It's estimated that intact coral reefs can mitigate 97% of wave energy. So when you have hurricanes in an era of climate change where we have stronger storms and more frequent storms, when that storm comes and you have these huge geologic calcium carbonate structures rising up from the ocean floor, it slams into that, mitigates 97% of the wave energy, and saves lives, saves coastal infrastructure. It's been doing this, kind of tapping out. Let me know if I need to reconnect to Zoom or reshare my screen. They support, because they support 25% of marine biodiversity, they support 5 to 15 tons of fish. So they are the primary protein source for hundreds of millions of people around the world, particularly in developing nations, around tropical or low latitude regions. And then they also generate around $36 billion of tourism revenue every year, because we love to go, we love to snorkel, we love to dive, etc. So if you wrap all of those ecosystem services up, it's almost $3 trillion in goods and services that these systems offer us each year free of service. And you would think with something that valuable, that iconic and irreplaceable, that we would do everything in our power to preserve them and conserve them. And unfortunately, they, as you know, are experiencing unprecedented rates of decline. And we'll talk about why that is. But first, a little Coral 101 is, and I apologize if this is rudimentary, but we get asked a lot whether they're a plant, animal, or a rock, and they are all three of those things. So if you look at a coral up close, this is one species of coral. What you have is a calcium carbonate rock, much like you have a skeleton that's covered with a really thin veneer of animal tissue. And inside that animal tissue is a symbiotic alga. And so there's an alga inside of it that provides upwards of 90 to 95% of its energy reserves through fixed carbon translocation. There's the animal tissue itself, and they're colonial organisms. So every single one of those tiny little white circles is an individual polyp that is communicating with all of the other individual polyps throughout the entire organism. And then there's the calcium carbonate structure underneath. So this is a schematic, a cross section of just a single polyp. And you can see that it has a mouth, it has a gut. The calcium carbonate skeleton gets deposited underneath. The tentacles actually will grab plankton and food particles out of the water column and then ingest them and digest them and then translocate food elsewhere. And you can see that in a lot of cases, the animal tissue is transparent, and then the single-celled endosymbiotic alga that live inside the tissue are photosynthesizing and providing it with up to 90% of its fixed carbon needs and its energetic reserves. And that symbiosis, it's an obligate symbiosis in many cases, that it's the breakdown of that symbiosis that causes coral bleaching and a lot of coral mortality, which we'll talk about again in a minute. So this is a video that we took at the Pal Academy. These are little baby coral larvae and they're adorable and I love them. And you can see how tiny and how small they are. And so corals reproduce asexually. You can clone them. You can break off a branch, and like a starfish, the branch will regenerate a new coral. The coral will regenerate a new branch, but obviously that's a single genotype. Or they reproduce sexually, and there's a couple of different ways they do that. Most corals reproduce sexually through once a year spawning, and they release eggs and sperm into the water column that fertilize. generate larvae like this that then go and settle and metamorphose. This is a metamorphosed new recruit that's probably 24, 48 hours old right there. So it has its aboral end of the larva is loaded with receptors, chemoreceptors, photoreceptors that probe the environment and try to find a good place to settle. You can imagine that's a very deterministic decision. And so despite looking deceivingly simple, these organisms at that life stage are very complex. So they probe the substrate, find a place to settle, and then the aboral end starts to flatten out, the oral end starts to differentiate and form tentacles and a mouth. And then that will start to secrete, the aboral end starts to secrete calcium carbonate, which you can see starting to form under here. They secrete aragonite, and this is the mouth. But it doesn't have symbionts yet at this stage, so it's just the transparent coral tissue.
I think Zoom is still connected, but the slides are not connected.
Do I need to reshare?
Yeah, sorry.
That's it. Yeah. So this individual is probably 4 or 5 days old. And then that individual, which started from a single larva, will start to asexually bud mouths and additional polyps around the edge. So this started as a single larva. The primary polyp and mouth is at the center, and you can see there's 5 radial polyps around the side. They start to acquire symbionts from the sediment and the water that's surrounding them, and establish symbiosis, because up to this point they're lithotrophic and they're feeding primarily off of lipid reserves. So once they establish symbiosis, they now have an alternate source of energy. This is a lateral view of what started as a single larva that's now budding mouths. And then they continue to grow asexually like that. This is a larger recruit that might be a few months old, all the way up until you have an actual coral colony. This individual might be a few years old, right there. You have hundreds of different coral colonies, several different species that will build reefs over time. This probably took hundreds of years, but in general, 10 to 15 years for a reef, a healthy reef to recover from in terms of coral growth, back from a disturbance. And then over geologic time scales, these individuals form massive calcium carbonate geologic structures that can be seen from outer space, like the Great Barrier Reef. And so the fact, I mean, I've been doing this for 25 years, and the fact that the Great Barrier Reef and these structures that can be seen from space all start from those single little sesame-sized seeds larvae continues to still just be a source of amazement for me. So back to where we are. So it's unfortunately, it is estimated that we have lost 50% of live coral cover since the 1970s. And about 50% of nearly 50% of the estimated coral species, we think there's somewhere between 700 to 800 coral species, and about 50% of those are now looking at elevated risk of extinction. So there was a special report of projections that the IPCC released in 2018, if we're looking at global warming of 1.5 degrees and what the implications for coral reefs would be. If we cap global warming at 1.5 degrees, the projections were that we will lose 70 to 90% of remaining coral reefs. And if we edge towards 2 degrees Celsius, which is looking more likely, we're looking at losing effectively everything, around 99%. So this is very much a dire situation. There's a variety of different reasons why, local and global stressors. So local stressors include development. So here you can see an airstrip that has been built directly on top of a coral reef for tourism purposes and presumably commercial purposes. There's overfishing, over extraction that destabilizes these systems and puts undue pressure on them, and that's both subsistence and commercial fisheries. And pollution. Water quality issues, pollution are a huge local stressor. We have a team of mesophotic divers at the academy that's led by Luis Rocha, our ichthyologist, who they dive to 500 feet, hundreds of miles off the coast of continents, archipelagos off the coast of Brazil. And at 500 feet, they will find tires, they will find plastic bags. There's really nowhere in the ocean that is a refuge from human pollution. Oh my goodness. Restart soon. Sorry, guys, this isn't my computer. I'm not sure.
So there's a little bit of a lag. Yep. Yeah.
So local stressors, obviously, in addition to global stressors, so everyone's aware that this is the first time in human history that atmospheric CO2 levels are over 415 parts per million. And the consequences of that for the oceans are warming and also ocean acidification, which particularly for marine critters who secrete calcium carbonate shells or skeletons for structural support or protection, makes life very difficult. The elephant in the room right now for coral reefs is warming, associated with out of control CO2 emissions. And so this is showing temperature anomalies. We've already experienced 1.1 degrees Celsius warming since the 1880s, which is 1.9 degrees Fahrenheit. And because over 70% of the earth is oceans and water has a high thermal capacity, the oceans absorb a lot of that heat, and they have absorbed about 93% of the heat associated with these unmitigated CO2 emissions. And that's a huge service to us, obviously, because it means that otherwise warming would be extremely worse. However, it's not without consequence. We see increased rates of coral bleaching all around the world. Here on the left, we have a healthy reef. On the right, we have a bleached reef. We see pictures like this in the news all the time now. This was taken by Luis and his team. I believe it's in American Samoa or Palau, I forget exactly where, just a couple of months apart. So the capacity for these warming events to cause mass destruction over very short timescales for coral reefs is very concerning to the scientific community, especially because corals are such slow growing organisms. So in a healthy scenario, a reef can recover in 10 to 15 years from a strong disturbance event. But it's estimated that right now, these events are spaced about every 6 years apart. So we're just not giving them any breathing. So bleaching is a destabilization of this symbiosis. It's really a stress response. It can happen in response to any sort of stressor, salinity stress, cold stress, dark stress, heat stress. But right now, because of global warming, that's the biggest cause of mass global bleaching events. And a bleached coral is not a dead coral. If it can regain the algae if the stressor goes away, but if it doesn't regain that algae within a certain time frame, it effectively starves to death because that algae was providing it with 95% of its food. We have had isolated bleaching events probably since forever, but global mass bleaching events where you have the vast majority of corals and reefs worldwide bleaching at the exact same time. There have been 3 of those events. There was one in 1998, one in 2010, and one in 2015 that lasted several years. And that was by far the worst and most widespread bleaching event that ever happened. This is a heat map associated with that event, and you can see that there was no place in the world that escaped this.
So over 70% of reefs worldwide were affected by that event. The Great Barrier Reef, we started to see media like this. The Great Barrier Reef suffered its third mass bleaching event in 5 years. And if you need 10 to 15 years to recover, obviously the math doesn't add up. And sadly, this story is not getting any better. So just this year, you've probably seen about the marine heat wave that's affecting the Florida Keys and the Caribbean that happened this summer, where people were going out into coral nurseries and restoration sites and just frantically pulling corals on land into land-based nurseries to try to get them refuge from the heat wave that was happening. I read one report where they rescued, divers scrambling rescued 32 to 33,000 pieces of corals from restoration and nursery sites to try to just get them out of the water and get them through this heat wave so that they could then place them back. And so that ties into what I'm going to talk about next, which is the need for conservation breeding programs and land-based nurseries and conservation breeding efforts in conjunction with zoos and aquariums and places like the Academy. So we already talked about this. Mass bleachings now is occurring 5 times more frequently than was previously estimated. They're about every 6 years instead of every 30 years, which gives no time for recovery. And if that wasn't bad enough, the recovery processes that we talked about, which are growth and reproduction, are both pretty heavily impacted by ocean acidification for these critters and for other marine invertebrates. So it's a double whammy where they're getting these acute disturbances that wipes out everything or a lot of things, and then their processes to recover are also compromised, so that erodes away these systems' resilience. Okay, so we're going to talk about solutions and hope. There's a lot of people that are tackling this from a lot of different angles right now. This was a report that was released a couple of years ago by the National Academy of Sciences, talking about all the different solutions and angles that people are trying to come at this problem from. So there's some you can see on this list, managed selection, managed breeding, coral cryopreservation. We're creating germ banks for corals in biosecure locations all around the world to try to harbor genetic diversity, manipulating the algal symbiont populations because there are certain strains of algae that are known to infer more thermal tolerance to the coral host, antibiotics, everything. People are going through everything right now, shading them, cooling them. During the most recent heat wave, there were people on listservs posting how to go out and put shade cloth over your corals to try to mitigate any additional light and irradiance stress. As part of this, one of the areas of focus that I have really channeled my energy to since I've been at the Academy is helping to establish land-based efforts to establish corals both as model systems, but also to establish conservation breeding programs. You think about other endangered animals like gray wolves and things that in association with zoos and aquariums, giant pandas throughout the world, they've gone to AZA institutions. The Steinhardt Aquarium that is at the Cal Academy is an AZA accredited aquaria, where they establish these conservation breeding programs and establish standards and protocols for keeping these animals in captivity so that you can ensure their survival and perpetuity. So that, and there's a lot that goes into establishing that. It's not just pulling it in and trying to keep it alive. I mean, corals are notoriously fickle animals to keep happy in glass houses, which is why they haven't really made good model systems to date. But you also need to understand what happened. Sorry.
You also need to take into consideration all of the genetic winnowing. So how heterogeneous of a population do you need to start with? If you're breeding generation after generation, is there any selection in a laboratory environment? Are you, and if there is, are there implications for rewilding those individuals down the line, et cetera, et cetera? So, so when I started at the Academy in 2016, it was the first time that I had ever not been proximate to coral reefs. But inside the Academy, if you have been there, we have a world-renowned aquarium, Steinhardt Aquarium, that actually has the Philippines Coral Reef Tank, is the largest, the tallest indoor tropical coral reef aquarium in the world. And so I kind of just missed working with corals, and I went and I talked to the director of the aquarium, and I said, I'd really like to start a system where we look at trying to do research on these animals here. I feel like this is such a lost opportunity. Like, let's do research on these animals. Let's try to complete the life cycle. Like, we can do so much with a system like this, because there aren't that many institutions in the world that have the know-how of aquarium staff who are experts in terms of husbandry and keeping those critters happy in glass houses.
Yeah.
Is there anybody online stuff?
There.
I was gonna say. So usually, if you're in a university or somewhere, that the onus of keeping those animals alive, which is not insignificant by any stretch of the imagination, falls on the researchers. And we're pretty good at researching these animals, but we're not great at like keeping them alive long term. And so there's so much strength in partnering with professional AZA accredited institutions. And so there's a real niche for this. And I think also having the Academy embedded in a tech and academic hub like the Bay Area gives us unparalleled opportunities to do really cool stuff if we have access to all life stages on these critters, things that you can't do elsewhere where you don't have the same technology access or support. So this is a lab, for lack of a better word, that I started in 2017. Our first spawn was in 2018. The main goals of this effort were to, like I said, advance conservation breeding techniques, establish corals as a model system so we can advance fundamental coral research and understand these incredible animals before that opportunity is up, and then also use it as a test bed for techniques and tools and technology that we can port to the field to on-the-ground restoration efforts elsewhere, used through collaborations with incredible institutions like you guys elsewhere. So there's a lot of different cues that people think go into eliciting coral spawning. So coral spawning happens once a year. It's usually once a year. And it's usually in late summer, after a full moon, a few hours after sunset. Different species do different things, but in general, that's the recipe. Happens once a year. About 75% of the 800 supposed coral species reproduce sexually this way. So this is a good process to understand how to make this happen if we want to know how to keep these animals happy. These are all cues that have been proposed as being important. And the first person to do this, I will say, we cannot take credit for developing. We were doing, so Jamie Craggs at the Horniman Museum in London was the first person to develop these techniques. He advised us in 2018 to build our system. And to our knowledge, we were the first place in the US, the second place in the world to do this. Now he has systems you can just buy, and he'll ship them to you in a shipping container for like $50,000. And it has everything you need to spawn corals. But at the time, that was not possible. So anyways, the things that we think are most important, although this one we've recently decided is probably not that important, are seasonal changes in temperature. So we think it's the warming in late summer that triggers the maturation of the eggs. So they have to experience this summer, they have to experience a winter. So we have 10 years of historical data from the site where these animals came from. We have two species, a Cropera hyacinthus and a Cropera millipora. The hyacinthus came from Palau, the millipora came from Australia. So we have historical sea surface temperature data so that we're plugging in and the historical monthly mean temperature, and every single day it's changing iteratively, either to increase or decrease towards the next month. And then lunar cycles, they spawn after a full moon. So we have LEDs that have 8 different channels. So we're manipulating the intensity and the combination of lights that they're experiencing throughout the day to mimic sunrises and mimic sunsets, to mimic full moons and new moons throughout the course of the month. And day length. So we plug all this information in: sunrise, sunset, moonrise, moonset, etc. And then the temperature and every single day they're experiencing. So to try to get them to think that they're in the wild, and it works. Which is super cool. This is a brief video to show you the process of building this facility. So it was a pilot project at first because we didn't know whether it would work. So we had to build a room within a room because light pollution was so critical. And then we sent a team to Palau, and they collected no more than a third of a colony and left the rest of the colony so that there was standing genetic diversity on the reef, shipped them back to San Francisco. We actually just received a shipment yesterday. We haven't gotten corals in several years because obviously the goal is not to take wild corals off of the reef. And then we just waited the first year and had no idea whether anything was going to happen. And they spawned, and it was awesome. And they all spawned synchronously at the same time that the wild colony spawned in Australia or in Palau. And it was super cool. And so now we have had 6, this is a cool video that a visiting student took. So I just show it because it was very cool. So we have done 6 spawns with 2 different species, and it has facilitated a wide range of really cool research, which I'm going to talk to you about. And we are in a very exciting phase where the Academy has deemed this a really important project and a capital project that this next year in 2024, they're going to expand and contribute a bunch of money to, which is really great, so that we can have multiple systems that are staggered, get spawning multiple times a year, and double the pace of scientific discovery. I swear this isn't me. Okay, so for example, I should have taken a photo of you because these guys are still alive and they're doing awesome. But this was a Cropera hyacinthus baby that was maybe like this big. I don't have a scale bar on it when it was 1 year old. This is 2 years old. And now we actually just moved them yesterday to make room for, and they're about this big. And they are 4 years old now. And we're hoping that they spawn and produce an F2 for us because we've sequenced all the F1s and we're looking at signals of selection in the laboratory environment. So I'm going to check these individuals for gametes in a couple of weeks to see whether they have any. This is how we check for gametes. You break the branch a month or 2 before the spawning and you can visibly see eggs in the branches. And we've gotten better over time. So this is our percent success rate of the individuals that spawned across a couple of different species. So every year we're getting a little bit better at this, which is good. You wouldn't want to go the other way. So we've done some cool stuff with this. One of the first things we did, we wanted to work, since we wanted to kind of establish it as model systems, is obviously you want a good genome. And so a Cropera millipora already has a genome. Hyacinthus didn't. And so we, in collaboration with Stanford and Dovetail, we created a genome for hyacinthus, and it currently is the most contiguous coral genome available. It's available for anybody. There's several research groups that are using it right now, and then we use that to ask some really cool questions. So this is Elora Lopez Vandam. She was a postdoc, she's now a research scientist in my lab. And one of the first projects that we worked on together was a kinship and selection question, looking at the differences genetically of all the wild caught parents and all of the 2 cohorts that we had of hyacinthus that survived to 1 and 2 years in our system, to see what parentage was like, what kinship was like, and whether there were signals of selection in the laboratory environment. So there were, we sequenced all the wild spawners, all of the babies, and started to build some pedigrees, and we wanted to do something like this. We need to sub the coral pictures in. It's not really quite as compelling when you put pictures of corals, but like it's still kind of. But these were the pedigrees for our 2019 cohort. So you can see that almost all of the babies that we had were full sibs of one another. And then there was one baby that survived. That was completely different. In 2020, we had all of these spawners down here that had nothing that survived. They spawned great. Nothing survived from them. And then we had a little love triangle over here where you have 4 full sibs and then and a couple half sibs. And then this individual over here, that was the result of 2 separate parents. So we built pedigrees, and then she started to scan the genomes to look for single nucleotide polymorphisms that were present at different frequencies in the babies than in the wild parents, and found a subset of 652 SNPs that indicated that there actually is selection happening in the laboratory environment. We collaborated with Molly Schumer's lab, a PhD student named Cheyenne Payne, who then looked at what genes those SNPs were associated with and what pathways. And it seemed like most of them were associated with vesicle transporting and functions that seemed to implicate symbiosis. So was some indication that there's some selection happening in the laboratory environment that might implicate symbiosis. And that's something we're chasing up right now. We had a bioinformatics undergraduate at Iowa State who took the WGS data, the whole genome data, because when we blast, about 80% of it was coral and about 20% of it was other stuff. So he took the other stuff and tried to see whether there were any ITS2 reads in there, because those are typically what's used to identify the algal communities and characterize those. And he was able to identify ITS2 reads in that data, and then looked at whether they were different in the parents versus the siblings, again, to kind of look at this question of symbiosis. And we need to validate these results by actually doing ITS2 sequencing of them, but based off of that data, this is just the PCA of the results, and the parents, all the wild caught parents clustered pretty closely together, but the babies don't. So these are the 2020 babies, and these are, most of them are over here, the 2019 babies. So it does look like the symbiont communities of the babies are different than the results, and they're also different between cohorts, which is interesting. So understanding that is one of the questions that we're chasing up at the moment. So TBD on the end of that story. Another really cool study that we did, having access to different life stages, is we were actually able to track mutation inheritance. So we could break branches. What we did was break branches off of these and put them in individual, tiny little branches, put them in individual tubes, and then that branch actually still spawned. So we knew that those sperm and eggs came from that adult tissue. And then Elora sequenced and looked for at mutations in the sperm and the adult tissue, just look at inheritance to see how many. This was to look at whether or not corals have designated germlines or not. and what that might mean for adaptive potential and inheritance of mutations. And what she found was that some are and some aren't. And so it kind of led to this hypothesis that there's self-regenerating stem cell lines in corals that regenerate throughout the lifetime of the coral that at any point can differentiate into germ or soma, and that the mutations in those stem cells can increase the mutation load of the sperm and the germ cells by up to 66%. And you can imagine that that would implicate adaptive capacity of these individuals in a time of rapid change and the potential for inheriting potentially beneficial traits. So we're chasing that up a little bit more as well. Is this end at 4:30? So this is a map of, so those are studies we've been doing in-house, because we do have at the Academy being a natural history museum, is very well equipped to characterize species and biodiversity. So we have an extensive microscopy lab, and we have an extensive genomics facility, Center for Comparative Genomics. So we've done that work in-house. And then we've had a lot of really cool collaborations externally as well, a couple of which were with professors here from Berkeley, which is really exciting. So just to highlight a couple of these, we've had one behavioral imaging study that has been running for several years now with Manu Prakash's lab at Stanford, where he's able to image these corals in 3D at microscopic levels and keep them swimming. It kind of rotates, and then you can introduce cues and see how it changes the morphology or the behavior of the corals and track it in 3 dimensions. So we have looked at coral larval behavior in response to temperature exposure, in response to different isolated compounds to see what might trigger settlement or a change in swimming behavior in response to different wavelengths of light. And Ian Ho, who's the PhD student that's working up that data, has a couple, I think, of pending publications on that. And we have another fun one is with the Shedd Aquarium in the University of Hawaii, where we're looking at the metabolomics of coral spawning, so we can sample the water that these corals are in. The fact that they all spawn synchronously once a year has kind of always eluded everyone. Like, how do they communicate with one another? What's going on with pheromones? And there's people that have studied this and gotten to various degrees of uncertainty around it. And so we started a metabolomics study that's still in progress, sampling the water before, during, and after the spawn to try to understand what some of these signaling compounds might be that actually contribute to the synchronicity of these events. And being at a museum, we've done a lot of really cool outreach too. So during COVID, we still have a live stream camera that's hooked up. So anytime the corals spawn, you can sign into this link and see them spawning, which is really fun. And then we do some cool engagement and outreach. So this coral spawning right there, we can hook into like elementary school classrooms and show people things, which is really fun. And just bring awareness to this issue. So I already mentioned this, but we are expanding at the moment, not yet, but hopefully the commitment's there. So hopefully by 2024, December of 2024, we'll have a dedicated room with dedicated research space, 2 systems that are staggered, and we'll be able to facilitate more research. And then we also were recently awarded a CORDAP grant, which is a funding agency that was a result of the G20 with the intent, the mission to accelerate restoration science and our ability to restore these ecosystems at the pace of their loss. So we have a big hurdle right now that when it comes to restoration science and restoration practices, we're pretty good at restoring reefs on the scale of like a hectare, but we're losing them at much greater spatial scales than that. So there's a lot of focus right now on scaling restoration techniques. And so we were awarded, a grant that just started last week, I think. So it's gonna run for 3 years, and it's in conjunction with the Roatan Marine Park in Honduras, and we're gonna be building a facility like this down there on Roatan. And then the goal is to test interventions to enhance growth and survivorship of coral recruits, so that when natural spawning events happen, we can help these sexual recruits. Specifically, we want sexual recruits so that we can enhance genetic diversity in the face of global change. Help them grow and help them survive better, and do that at scale. So we're testing, there's 4 techniques that we're testing up here, and these are super simple, but they're designed to be super simple so that they're accessible and easy to implement at scale. So they're buffering the seawater, and part of this is stemming from my OA background, but hardening is a practice in some shellfish industries to help early life stages calcify more quickly, and give them an edge. And we know in coral recruits that survivorship is size dependent. The bigger you are, the better you survive. So if we can help them calcify more quickly by increasing the alkalinity of the seawater in those early stages, perhaps that translates to better survivorship. And we trialed this last year, just piloted it, and there were this was actually unexpected because this is not the growth data, this is just the number of recruits. So the number of settlers was 100% higher in the buffer treatment than in the control treatment, which I haven't quite figured out why that would be yet. I would expect it to enhance growth and calcification, but not actual settlement, but I'll take it. So we're chasing that one up this year. And then we're gonna dose with amino acids and symbionts, and then we're doing a heat selection experiment. So this is Elora's idea. This is us trialing this one last year. And this is to try to see if we can select for thermally tolerant larvae, with the idea that at that stage, you have as much genetic diversity as possible. So we exposed them to 36 degrees Celsius. The controls were at 26 degrees Celsius, ramped it all the way up to 36 and tried to kill them. Not all of them, but we wanted to kill some of them. And we killed only about 5%. They're pretty sturdy at that stage. And then we wanted to grow those out and then stress them again 6 months later and see if we actually selected for individuals that were thermally tolerant. And we don't have all of the data to show you, but what this shows you is that, what does this show you? This shows you the percent of larvae surviving at the heat ramp. So showing you that controls that we did actually kill some off. So there was some sort of a selection event. And then this is the number of live recruits 5 weeks after settlement. So there were fewer on the heated. So we did, again, kill some off, which is what we were trying to do. But then 5 weeks later, survival, there was actually, this was not statistically significant, this was a pilot, but there were actually higher survival in the heated than in the controls, which was interesting. So we're gonna chase that up this year too. And we did single cell sequencing in conjunction with Cassandra Duran, who's at NASA Ames, of those individuals, of the larvae and the recruits, to look at transcriptomic signatures at different life stages. And we saw no real difference of control or heated larvae at the larval stage. They don't really seem to care, according to this, which it kind of tracks with what we saw. We just heated these guys up. We kept saying like 32, let's try it, 33, nothing, 34, nothing, 35. Like they're tough. And then the settlers, there's really only one conserved group of cells. So the settlers, there was much more differentiation, like transcriptomically, in terms of what was going on. And obviously left to right is the developmental shift that you would expect anyways. And the goal is to try to identify, see if any of these techniques work. And then if they do, port them. So 3 of our Honduran collaborators are coming up next month. Our spawn happens in December every year. So our next spawn is going to be December 10th. And the goal is they're going to help us run these experiments, and we're going to see if any of these techniques work. And if they work, we're building this facility in Roatan. We're going to take them down there, try them in the field with on the ground restoration practices. And then if it works, then hopefully try to integrate it at scale and help get more sexually diverse or genetically diverse corals out there that survive better. So this is just some of the things. So you can do so much with a system like this, and it has been a labor of love, but we're super happy to have it. So we're assembling reference genomes, looking at mutation inheritance, looking at which parents or genotypes are most successful, if the lab selection, if there's any signature of lab selection, if you can have interventions to increase growth and survival, translate that to on the ground practices, selecting for heat tolerance, et cetera, et cetera. These are some of the things that we're doing with this system. And this is the last slide that I wanted to show you, which is just spawning in the wild, just because it's so cool. This is a coral colony in That's terrible time. I'll show it. So this is spawning. One of our aquarium biologists took this. So you can see, if you were to slow that down, you'd see this wave across the whole colony. All of those individual coils are communicating with one another in ways that we don't understand. And releasing eggs and sperm. Those are gamete bundles, they're hermaphroditic. There's a sperm packet in the middle and it's surrounded with eggs on the outside, 5 to 7 eggs, depending on the species. And then that floats to the surface, breaks apart, sperm and eggs mix, fertilize one another, and then creates larvae. And this used to happen synchronously at scales where you would get, it still does some places where you get slicks of coral gametes, and there's these incredibly successful reproductive events, and it's breaking down over time. You can imagine as we lose corals, these populations become so depauperate that one might spawn, that one might spawn, they're never going to meet another ever. So we're trying to figure out ways to support this natural process. And that's it. I put a couple for people who are interested in any additional resources. I put some links up here to learn a little bit more. And I put my email if you are interested or have any questions, please feel free to reach out at any time. We're in Golden Gate Park, so not that far away. Thank you.
Thank you so much for that. I think given that we have any time for one question, and then we'll take the rest of the questions outside to the swimming lab.
So I'm struck by some of the efforts that folks say like in Florida are taking to remove corals from environments where there's heat. And I'm interested in what your thoughts are, because in Florida there's also the disease, the chronic tissue coralopathies, or something to that effect. In doing that, like, would you, as a museum curator or as an aquarium curator, be concerned about the threat that might pose on like other adjacent systems that may be within the similar water capacity of those?
Bringing diseased corals on
Yeah.
site.
Yeah.
Yeah, absolutely. I would assume that they have a protocol in practice where they would isolate them or treat the water. That's something you can put ozone or things in line that treat the water that come out, or what we do when we bring our corals in when we get shipments of corals, we have to dip them in a variety of different chemicals before they go into any tanks. And then our water is treated either with ozone or there's a variety of different options you can do just to make sure that you don't contaminate anything else.
That's another question.
Yeah, sure.
Okay, so for the new spawning systems that you have, one thing that, and if you're working with coral spawning, it's really tough is that they spawn at night, and then if you have to do a bunch of work with them now that they've spawned after the sunset, you're working through the night. So you mentioned that you're gonna get a new system to kind of like see if you can offset some
Yeah.
of that stuff. Is it advantageous from your perspective to be on the same schedule as the spawning that's happening, or like that's occurring naturally? Or is the goal to eventually make them happen more often? Like, are you gonna try to mess with that, or are you worried that you may break the system then?
So we shift our day-night cycle to your point, so that it's convenient. So our sunset, our corals spawn at 2 in the afternoon because our sunset's at 11 in the morning, because I don't want to be up at 3 in the morning. So that I think we're gonna keep, because I think it depends obviously on what your question is, but for us, we're always interested in like the timing of the spawn relative to the days after the full moon. And what a lot of ex-situ spawning systems have seen is a lag on the order of anywhere from 5 to 7 days. So our corals consistently spawn 12 to 15 nights after the full moon, and in the wild they usually spawn 5 to 7 nights after the full moon for these species. And that's true of other systems in other countries too. So I don't know what that lag is about, if they're jet lagged literally, but the timing of the spawn during the day, we don't have any scientific questions where that would be important for us. It's more just how much we can get done before getting too exhausted. In terms of truncating it, it's something we've thought about, and it's always a question because you only have, right now we only have the one system, and when we get another one, we only have the 2 systems. So how much you want to play with it and risk not getting anything is always something to think about. But I have had some conversations with people at Hopkins that there had been some studies in other marine inverts, I think it was in Ascidian, where they truncated the photo period. So instead of a 12-12, they did a 6-6. And in 3 generations, that individual started spawning twice as quickly as it would have previously, which makes you wonder if we did that in coral. I mean, 3 generations for an Ascidian is like 3 weeks, but 3 generations for a coral here, it's years.
Yeah.
So I have wanted to do that, but I don't want to wait 3 years to figure out whether it works. But it's something someone should do, because that would be great if you could expedite things, but then you run into also needing to validate whether there's any implications of the gaming quality if you're rushing things. So obviously you'd have to like make sure. But yeah.
Let's take the rest of our questions outside and thank you.
And if you have any questions about OA or OAE or any of that stuff, OA policy, I'd love to chat with you too, because that's also near and dear to my heart, and maybe I can come back and chat about that sometime. Thank you.